6 base pairs, heme occurs at a particular point. Okay, so after 6 base pairs, understood? Okay. Next, the cofactor of the enzyme carboxypeptidase is, for carboxypeptidase, the cofactor of the enzyme is zinc. Okay, for carboxypeptidase, zinc. And for niacin is associated with NAD and NADP. Okay. Next, which of the following are required for dark reaction of photosynthesis? So for dark reaction, what is needed? Light is for during daytime, light, so not needed. Chlorophyll is also for during daytime. And CO2, yeah, ATP, NADPH, all these are needed to fix the CO2, and it happen during dark times. So C, D and E. Okay. Next, the type of conservation in which the threatened species are taken out from their natural habitat and placed in special setting where they can be protected and given special care. So taking the species from a particular conservation, from a particular their natural habitat, and placing them in the other place, other special setting which is not real. So what is that? It is called biodiversity conservation. We are conserving the animals to not become extinct. Okay, it is called biodiversity conservation, and it is where species are taken from natural habitat and placed in a special setting. Okay, and this is called ex situ conservation. They are taking out and placing at the other place, ex situ, and this ex situ comes under biodiversity conservation. Okay, next, again matches. So two or more alternative forms of a gene. Two or more alternative forms of a gene is called allele. Yes, 3. Cross of F1 progeny with homozygous recessive parent. Okay, with homozygous recessive parent, F1 cross is called test cross. Okay. 4. Cross of F1 progeny with any parent. Any parent is just back cross. Okay. 1. Number of chromosome sets in a plant. Chromosome sets. It's called ploidy. Chromosome sets. So 3, 4, 1, 2. 3, 4, 1, 2 is option 2. Okay. Next, formation of interfascicular cambium and fully developed parenchyma cells. Okay. So when interfascicular cambium is formed from fully developed parenchyma cells, this is called dedifferentiation. Okay, dedifferentiate. From already developed cells, this cambium is forming, interfascicular cambium. It is called dedifferentiate. Okay. Next, this phenomenon is dedifferentiation. Spindle fibers attach to kinetochores of chromosomes during which phase? During metaphase stage only, this kinetochores of chromosomes attach to spindle fibers. Okay, metaphase, at first phase, kinetochores attach to spindle fibers. In a plant, black seed color is dominant over white seed color. In order to find out the genotype of the black seed plant, with which of the following genotype will you cross it? Okay, suppose to determine the gene type of a black seed color at F2. Okay, the black seed from F2 is crossed with white seed color. It is black seed, and the white seed it is crossed. It is called test cross. You are crossing it with homozygous, so it is called test cross. So to determine whether it is dominant or the genotype of the black seed plant is dominant, you have to cross this with recessive. Okay. Next, a pink flower snapdragon plant was crossed with a flowered snapdragon plant. What type of phenotype is expected in the progeny? So pink color snapdragon plant genotype is RR. Okay, pink one. Next, and red flowered, here it is given, not given red, so red flowered. Okay, So if they both are crossed, let's see the cross. Okay, here dominant, here pink, here red. If you cross R, so 2 are red, 2 are pink, 2 red and 2 pink. Only pink and red flowers are there. Red as well as pink, right? Only pink wrong. Red, pink, white wrong. Only red wrong. Both red and pink are there. Okay. Next, inhibition of succinic dehydrogenase enzyme by malonate is a classical example of. So this enzyme malonate shows close structural similarity with the substrate and also competes with the substrate for the substrate binding site of the enzyme. Succinic dehydrogenase means this malonate appears similar or same to the substrate and manipulates to go for the substrate binding site of this enzyme. Okay, it is willing to reach this enzyme. So it is called as, it is competing with other, it is resembling other to go for substrate binding site. It is competing with others, so it is called competitive inhibition. Okay, next, given are two statements, so let us see which of them are right. Next, Bt toxins are insects group specific and coded by gene CRY1AC, right? And is it right? Yes, right. Bt toxin exists as inactive protein in Bacillus thuringiensis. However, even after ingestion by the insect, the inactive toxin gets converted into active from due to acidic pH of insect gut. So, as Bt toxin were isolated